So this chapter is on how cells harvest energy, and this is basically how cells are going to take an energy source, whether it's an organic molecule or inorganic, and they're going to use the energy and the bonds in those compounds to make ATP. And ATP is a universal currency that cells use to do all of the work within a cell. So our first section here is an overview of the process called cellular respiration. And your learning outcomes are to characterize the oxidation dehydrogenation, which means to remove a hydrogen, reactions that occur in biological systems. So basically, the form of oxidation is going to be a dehydrogenation. So we'll go over that. And then explain the role of electron carriers in energy metabolism. We'll go over some electron carriers, and then we're going to describe the role of ATP in your biological systems. So when we look at organisms, we can classify them based on how they obtain their energy. Okay, so or their mode of nutritional mode. Sometimes they might say it. So you have autotrophs and then you have heterotrophs. And autotrophs, like automatic, these are, they feed themselves pretty much. So these are capable of making their own organic molecules. And those organic molecules have lots of carbon-hydrogen bonds where we get our energy from. And one big group of autotrophs are the photoautotrophs, which do photosynthesis. There's also chemoautotrophs, and then we can subdivide those into further categories. But essentially, the ones we're most familiar with are the ones that do photosynthesis. And then we have heterotrophs, and heterotrophs include animals, like the human animal, and other organisms like fungi. They cannot make their own food, so they have to rely on organic compounds that are made from other organisms. But all organisms, regardless, whether you're autotroph or heterotroph, are going to use cellular respiration, which we might define as a way, the processes that are used to extract energy from those organic compounds. So the organic compounds be like glucose, for example. So cellular respiration generally is going to be a series of reactions. And these reactions involve both oxidation and reduction. And recall from our earlier chapters in chemistry and even in the prior chapter just on general metabolism, that when you lose electrons, that's called oxidation, and when a compound gains, that's called a reduction. Since we cannot create or destroy charged particles in normal reactions, you cannot just create electrons. They have to come from somewhere. So something has to lose if the other is going to gain. So these have to occur in pairs. Now, the major type of oxidation we're going to be considering here involves the loss of a hydrogen. So we're going to call that dehydrogenation. And dehydrogenation is often facilitated by enzymes called dehydrogenases. And here, basically, something similar that I mentioned in a prior lecture, that when it comes to following electrons, when it comes to these reactions involving organic compounds, follow the hydrogens, right? So in a prior lecture, I talked about, let's say we have an organic compound, which is going to symbolize with R, just represents a larger molecule with carbon, hydrogen, oxygens, and so on. It could be glucose, for example, and it has a hydrogen on it. Okay, and all we have to do is basically follow where the hydrogen is going, and wherever the hydrogen is going, it's taking its electron with it. So when we dehydrogenate, we would be removing the hydrogen, and we could add it to another organic compound. We might add it to an inorganic compound. It's just but you follow that hydrogen. And we'll see cases where we're going to add it to inorganic, like oxygen, for example, to make water. But just R prime just means some other compound, organic compound. And so the compound number 1 lost the hydrogen, so it was oxidized. And the second one, R prime, gains it. And not only did it gain it, it gained it with that electron, so it's going to be the one that's reduced. The hydrogen and the electrons that are right there, there's a lot of energy in that bond. So wherever those electrons are going, the energy is being transferred with those electrons. Okay, so remember that a hydrogen atom basically is one proton with one electron. So we're moving that the electron with the hydrogen. So some compounds got to gain, some's going to lose it. The redox, the oxidation reduction. Remember that the way we're going to oxidize for these organic compounds is with the removal of hydrogen. Sorry, so when we move the hydrogen, we're going to remove the electrons with it, right? And so they're called redox reactions because they involve both. And where might we take that hydrogen? There's going to be a group of compounds called electron carriers. And some of them are free to move about, and one of them is called NAD+, and that's short for nicotinamide adenosine dinucleotide. Remember that nucleotides are what we find in DNA, right? Like there is a nucleotide that has adenine in it, which is one of the nitrogenous bases. So, but this one's a dinucleotide, which means it's two nucleotides together, and it has a positive charge to it. And basically, this compound is going to take electrons from organic compounds. We're going to transfer the electron onto this one with the hydrogen by oxidizing the food particle. It might be glucose, for example. And right now, the NAD is in its oxidized form. Okay? And so if we were to take, say, that compound from earlier, and we remove that hydrogen, okay? And we move that hydrogen on here with a couple of electrons. And hydrogen, when it loses an electron, is a proton. So we take that proton and we add it to this. It has a positive charge. The NAD has a positive charge right here. And then if we were, so we're going to now have a +2 charge, but if we add 2 electrons, which can come from your food particles, then what you end up with is your NAD, the plus, you're going to put the hydrogen on there. Right now that's 2 pluses, but if we throw the 2 electrons on there, we now have NADH with no charge because it's now neutral, and this would be the reduced form. Okay. This NADH then can take the electron that it's carrying along with that hydrogen and the energy in that electron to other processes that are going to be used to make ATP, right? So for now, we're going to know NAD as one of those major electron carriers. We're going to see it come about, right? And so let's take the diagram here. And when we follow this, when we oxidize one, another compound has to be reduced. And right here, when they say oxidize, they're referring to your energy-rich compound that's right here. In this case, it's our organic molecule. And they just drew it as an oval shape. It's just some carbon molecule, could be glucose, for example. And here are some couple of hydrogens on there. This is an enzyme that's going to facilitate the reaction. Okay, it's going to bind with your energy-rich organic compound, and here is your oxidized form of NAD+. So binding together, then the enzyme facilitates the transfer of the proton with those electrons to the NAD+. Okay, so what are we doing? If we're dehydrogenation of the organic compound, we're oxidizing it because we're removing the hydrogen with the electron. And we're removing it, and we're going to add it to the NAD+. So we see that going on here. This is the process going on right here. You can see we're going to transfer onto the NAD+. We're going to transfer one of the protons, and we're going to take 2 electrons off of there to balance. And if we do, each of those hydrogens has an electron. So we're going to take both electrons. There's one for this hydrogen, one for this one. We're going to take both electrons and one of the protons and add it on there. And what's that's going to leave is going to leave 1 proton that's just going to be there dissolved in the environment. And what ends up happening is now we have our products. They're released just like when we were covering enzymes in the prior chapter. And here we have our reduced form of NADH, which now can be used to take that high energy electron to go do some other work, in this case to make ATP. Okay. Now, if we were to take this organic compound and go the other way as a reduction, we would have to add the hydrogens back on that, but that's not very favored in this case. When we're oxidizing organic molecules, they have a lot of free energy in there, and the free energy change is going to be negative. Okay. And that means it's a spontaneous process. It's facilitated by the enzymes like we covered in the last one. But when we say negative, that means energy is given off. And where's that energy given off to? Well, it's going to be basically a lot of that energy is going to be placed on and carried in the reduced form of NADH. So it'll take that energy elsewhere. Now, remember, when we transfer energy, according to thermodynamics, second law, we're going to lose some energy in the form of heat, right? So not all of the energy is transferred there. Your metabolism generates heat. But some of that energy that's lost from your organic compound is actually going to be held on that NADH, and that energy is going to be carried to go make ATP. Okay, so, but if we were to do the other and go the other way, that's not favored. The reduction of that carbon compound would be positive. And this is where autotrophs come in, like plants, which do photosynthesis. They're going to take the energy from the sun to drive the reaction the other way to make back their energy-rich compound. Okay? But the key thing to remember here is what's happening here. We're going to oxidize the organic compound by removing the hydrogen, which is going to take that electron off with it. That's on oxidation. What are we going to do? We're going to take that hydrogen and the electron, and we're going to put it on an electron carrier like NAD+. Okay? And when we do that, we oxidize the food compound. We're going to oxidize your organic compound. And you're gonna oxidize this one, and you're basically going to reduce NAD to make its reduced form here.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron, a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
This process is not going to occur all in one step. If not, too much heat would be generated. So we're going to do this over many, many steps. So there's going to be dozens of these oxidation reduction reactions. Okay? And in many cases, a lot of these reactions are going to use electron carriers like NAD+. There's another one called FAD, which will also carry hydrogens with electrons. And they're going to carry these high energy electrons to go do the work of making ATP. Okay, so ultimately, those electrons that are being carried with these electron carriers have to be transferred onto a final electron acceptor. And the kind of acceptor depends on what kind of respiration we're talking about. Is it oxidative respiration, which we're going to be using oxygen as the final electron acceptor. In which case then, if you have one oxygen, half an oxygen molecule, it would be just an oxygen atom there. We can take the protons off of there and add them here, and then the two electrons and add them there. What we get is water. So when the oxygen is your final acceptor, you end up getting metabolic water at the end of your metabolism. So we'll see water appear there in a bit. There's also anaerobic respiration. So I mentioned you have aerobic respiration, and here your final acceptor is going to be oxygen, and that's in which case we make water at the end. The hydrogens get added on there. So if we take your oxygen molecule is basically like this, following the octet rule, and if we break it apart, you get two unstable oxygen atoms with their dot structures. And if we just throw those electrons on here, one there and one there, and then attach two protons, which are hydrogen atoms without their electrons, you get your water molecules. So the end result here is you get water at the end. So if the hydrogens are ending up on your oxygen, then what are we reducing at the end? We're reducing oxygen, and that makes water. Okay, so then we have anaerobic respiration. Here, final electron acceptor it's not going to be oxygen, something else. And it may be inorganic or organic. So if it's inorganic, we'll just refer to it as just plain old anaerobic respiration. And your final acceptor might be something like carbon dioxide or something else that's not considered organic. When it's fermentation, it'll be an organic molecule. And so we're going to see examples of fermentation a little bit later. In one case, you end up making ethanol, which is the alcohol you find like in wines and beers and so on. In this case, your final acceptor ends up producing those compounds at the end. So when we look at aerobic respiration, and we're actually using oxygen. So we see oxygen here in this formula here. And this is C6H12O6 is familiar. This would be something like glucose. So basically, this is your organic molecule. And your organic molecule has hydrogen on there, and there's the hydrogen with the electrons. We're going to dehydrogenate. We're going to remove the hydrogen with its electrons. That means you're oxidizing your organic molecule. And then your oxygen is going to be your final electron acceptor. Ultimately, you're going to produce some carbon dioxides, and because the oxygen is going to be your electron acceptor, every oxygen atom will end up picking up 2 of those electrons that came from the food molecule with 2 positively charged protons on there to make water. So that's where you get your water from. Okay, so your oxygen, the oxygen basically becomes reduced to make water. Okay, so here's your metabolic water here, and what you're going to end up with is energy here. Now, the free energy change. Okay, remember free energy change. You're going to have a certain amount of free energy before and a certain after. It's going to be negative. Now, we know that from the last chapter that makes this exergonic. And exergonic also means spontaneous. Now, if all of this free energy would be released at once within your cells, you would burn up, right? So we cannot do this all at once. It has to be done under controlled steps. So you're going to have enzymes with those dozens of reactions that we were talking about in the prior slide, dozens of reactions where we're transferring those electrons from one molecule to another, ultimately ending up on oxygen to make that water. And so this is going to be done in smaller steps rather than all at once. And so they use this analogy here. So we start with your food molecule here that has the hydrogens and the electrons on there. And these are your electrons that have been pulled off that food molecule. They're going to start off with very high energy. And your analogy here is as you're transferring those electrons with their hydrogens, you're going through steps. And as you're going through steps, what's happening to the energy of those electrons? They're going to lower and lower free energies, right? We start off with a high free energy. Remember, we have a free energy is going to be high. And then over here, your free energy at the bottom is going to be low. So that means the change in the free energy is going to be negative. Well, how negative? Minus 686 kilocalories per mole of glucose, right? So where's that energy going? Some of that energy, not all of it, because the transfer is not 100% efficient, is going to be going to make ATP. So we're going to use some of that energy as we're transferring, some of that energy is going to be used to make that ATP. Where does that those electrons ultimately end up with? Well, oxygen is never by itself. It's a diatomic molecule. It's always found with another oxygen atom. But the processes break apart the oxygen to make one radical form of oxygen, which is not very stable. So that's why they say half an oxygen in there. So half of this is an oxygen atom. You're going to take 2 protons from the environment. Some of them probably came off the food molecule earlier. So it's just a mix of all of this stuff within the cell. And then your electrons ultimately end up with those 2 protons, the oxygen atom, to make your water, like was mentioned up over here. So it's done in steps. And we're going to go through those steps, right? There's going to be different types of electron carriers that are carrying these high energy electrons down those steps we were talking about from one molecule to another over dozens of reactions. Now, some of them are free to move around and diffuse and go through membranes and so on. So they're quite soluble. But some of those electron carriers are also going to be bound within membranes. So they're going to be within those membranes, within that membrane environment, and they too carry those electrons. So when they receive the electrons, they're going to be reduced. When they lose the electron to the next molecule, they're oxidized. So these carriers are reversibly oxidized and reduced. As they get the electrons, they're reduced, right, when you receive them. When you give them up to the next molecule, you become oxidized. So what we're doing is we're transferring those electrons as they go down the steps, okay? When it comes to some of those important carriers, recall the structure of the mitochondria. We all know the mitochondria is the powerhouse of the cell. We want to get away from just using that and start saying, hey, this is the place where we're oxidizing food molecules, organic molecules, in order to take that energy, extract that energy from the molecule to make ATP. So remember that your mitochondrion has an outer membrane here, and then it has an inner membrane. Okay? And then between here is the for in between membrane space. Okay? Intermembrane space. We need to recall the basic structure of the mitochondria. That inner membrane is this one right here. Okay? It's a phospholipid bilayer just like any other membrane. And embedded within the membrane there are these carrier proteins. There's several of them in there. Okay? And so they're going to be oxidized and reduced. Okay? And as they're doing that, we're going to focus on what's happening to the protons, which a lot of them have come off the food. We've pulled them off. We pulled off the electrons as well. Some of those protons are going to go on to NAD, our electron carrier, and along with the electrons and produce NADH, right? And so out here in this diagram right here, we have our inner membrane space. So this area right here is your inner membrane space. That's this space right there in the drawing up above. Below the membrane is your matrix, okay? The inside of the inner membrane is called the matrix, okay? And there's a bunch of enzymes in there as well. So what's happening here is, well, remember the NAD was reduced because we oxidized the food and it's carrying the electrons with that hydrogen. Those NADHs are going to now come over here, and they're going to deliver the electrons to one of your first carriers there in what's called electron transport. So these carriers are either going to carry the electron and hold it for a bit. So when we move the electrons on there, you've reduced this molecule that's embedded within the membrane. Okay. At the same time, when you've delivered it, now you've freed up NAD+, and NAD+ can go back and pick up more electrons from other food molecules. But what we've basically done is we've oxidized NAD. It's lost its electron, and that electron has moved on to this first carrier here. That first carrier is now reduced. Okay. And then this carrier then transfers it to other carriers within the membrane, and every time we're transferring, it's a reduction and oxidation, reduction, oxidation, reduction, oxidation. And what we're doing is we're going down those steps. Remember those energy steps, right? And well, where is that energy going? Remember that energy is going to help produce ATP, a bit indirect though, because every time these carriers are moving those electrons, some of them are just carrying electrons, but some are going to carry both electrons and protons. And protons are the hydrogens without the electron. You see a whole bunch of protons here within the space here. What's happening is, as the carriers are carrying the electrons, they're going to carry the protons from the matrix out here to the inner membrane space. So it's like we're pumping those protons with the energy from the electrons into this inner membrane space that's up here. And so what we're going to do is we're going to pack a whole bunch of electrons in that inner membrane space right up here, and you're going to build up this potential energy because you're going to build up this gradient of protons, which have both a positive charge plus their chemical potential energy of just being together. They're going to want to move away from each other, right? And so those protons, because they have a positive charge, can't just move through the phospholipid bilayer. Let me get rid of that. They're kind of repelled from there, because remember that the inside of a membrane is hydrophobic, doesn't like anything that dissolves in water. Okay? But we have a whole bunch of potential energy buildup in this inner membrane space up here, and it wants to diffuse back in, can't go through the membrane. So where does it go? It goes through special membrane protein right here. And that protein is called ATP synthase. And the ASE means enzyme, and synthesize means to make. And what is this enzyme going to do? It's going to make ATP. So all of this buildup of protons here, the energy used to build up those protons came from your food, came from the transfer, oxidation, and reduction, going down the steps here. We built up this proton gradient, which can now move back into the matrix through ATP synthase, and that kinetic energy is what's going to drive the production of ATP. And all you need on the inside of the matrix is your ADP, which is adenosine diphosphate, which is a used up ATP molecule. There's the two phosphates there. And then you're going to get an inorganic phosphate with the help of this enzyme to reattach the phosphate to make your ATP, which now can then diffuse away to go do the work of staying alive, biological process. So that's kind of it in a nutshell, and this is where the mitochondria becomes involved in it. Here's a picture of what NAD oxidized and reduced form here. On the left side is your oxidized form. And you can see it's got two nucleotides. Here's the ribose from one sugar. There's the other one. Here's your nitrogenous base adenine, which makes this one adenosine. And then here's the other part here. Here's your other nucleotide. But notice that the nucleotide is missing an electron. So the overall molecule has a positive charge. That's why we say NAD+. Okay? Now remember, we are going to oxidize food and get electron off the food, and we're going to take a hydrogen with the electron and attach it here. Now look at this position right here. Right there you have just one hydrogen. When we reduce the NAD, because we oxidize the food, we end up getting two hydrogens there. We've also put two electrons on there, which got rid of that positive charge that was right here. So this is your reduced form of what which becomes NADH. Remember that H has that high energy electron that came from the food. Okay? You don't have to worry about memorizing the structure, but it's just good to know, hey, that's what your molecules look like. But what you should remember is NAD+ is oxidized form, and NADH is going to be carrying that high energy electron in its reduced form. Okay, so when it comes to ATP, the ATP itself, when we're going to use it to power other processes, 1 mole of ATP. We're talking about moles and as a way of counting. So we have 1 mole of ATP if we do hydrolysis with it. So that means you're going to bring in water to split it. You're going to split off one of the phosphates to make ADP and then your inorganic phosphate. Okay. That process has a negative free energy. In other words, there's more free energy here, and then ADP has less free energy. So it's going to be a negative change. How much energy? Minus 7.3 kilocalories per mole. Okay. Now, if we're going to take the inorganic phosphate and ADP and go the other way, that would be a positive free energy change. That means we would have to put in free energy. And how much? It would be just the exact opposite of that. It would be plus 7.3. Where does that energy come from? We get it from the food. Okay, so the food, the electrons we're pulling off the food is going to be used to remake your ATP. So there's going to be two mechanisms for making ATP. One of them is substrate level, and then here's a fancy word to know: phosphorylation. Phosphorylation means when you add a phosphate onto a molecule. Anytime you add phosphorylation, that means you're going to add phosphate, an inorganic phosphate, to a molecule. Okay, and this one of the substrate level phosphorylations can occur when you take an ADP and an inorganic phosphate added on there, and we're going to see that process occur in several places along the way of breaking down the food. And one of them is called glycolysis. When we first split sugar and break it down, no oxygen is required, and it's going to be substrate level. Remember, enzyme substrate, right? Enzyme substrate complex. And so there is a diagram of substrate level down here. Okay. And if we go back earlier to so this occurs even before we get into the mitochondria. If we go back to the mitochondria picture here, this process right here, erase some of this here, overall is going to require an oxygen at the end. Okay. That oxygen is going to pick up the electrons that were delivered here. The electrons are going to come over here, 2 of those electrons with 2 protons to add with the oxygen to make our water here. When your final electron acceptor there is water, we're going to call that oxidative phosphorylation, because ultimately we're going to use that process to make more phosphates. And we're going to phosphorylate ADP over here. That's your oxidative phosphorylation, but we have phosphorylation occurring at the substrate level in processes before we even get to the mitochondria, right? So the oxidative phosphorylation is going to occur. This is going to occur in the mitochondrion. And mitochondrion is the singular form. Mitochondria is plural, right? So in this case here, substrate level, which is the actual process, one of the processes that do occur during glycolysis, we make a few ATPs there. there's an organic compound called phosphoenol pyruvate, and it has a phosphate on there. Okay, and that phosphate, with the help of the enzyme, is going to take an ADP, so it binds to an ADP, which has lower energy, adenosine diphosphate, and we're going to take the phosphate off of the phosphoenol pyruvate and attach it there. This is substrate level. Okay, there's no oxygen involved here. So once we're done with the reaction here, we've released the compound. Phosphoenol pyruvate just becomes pyruvate. PEP stands for phosphate because it has a phosphate on there. When we transfer, we get our phosphate. That goes, and we have our ATP that's been released, and that ATP can be used to go power other processes. So here, we're directly transferring the energy from an organic molecule onto ADP. When it comes to oxidative phosphorylation, we've removed hydrogens, and we're going to use those electrons to that electron transport on the ATP. In the mitochondria to do the phosphorylation that involves that enzyme ATP synthase. So the overall overview of everything that we're going to go over in the rest of the chapter. The first section is pretty long because it gives the background for everything here. If this is the mitochondrion here, okay, there's your inner membrane right here, okay, your outer membrane, the inner membrane space here, then out here would be the cytoplasm, okay. And so if I would recommend anything at all on how to remember a lot of the stuff, draw this diagram here and then put little spaces in front of these compounds so that you can write down how many of each you get. So including in front of the ATP. How many ATPs do we get from a process called glycolysis? And so glycolysis is the first process. Lysis means to split. Split what? Split sugar. Where does that occur? It occurs in the cytoplasm. They've drawn it outside of the mitochondria. Okay. When we're done, you're going to end up with pyruvate. Pyruvate enters in there, goes through an oxidation. That's the second part that happens as we're going into the mitochondrion. Okay, and actually you're going to end up with 2 pyruvates there. You're going to end up with so many NADHs. So what you're going to do is you can redraw this and then track how many of each you get. And when you're drawing, you're doing that kinesthetic learning that helps burn it into your mind, right? So when we're done, you can go back and fill in those numbers there, including how many ATPs are produced by Krebs cycle here, which look at the location of Krebs cycle. Okay, the Krebs cycle is also called the citric acid cycle. It's the same thing. There's a third name for it called a tricarboxylic acid cycle. So there's 3 names for it, but it occurs where? Well, what's that space on the inside of the mitochondria called? It occurs in the matrix. So you need to also know where these processes occur. Where does glycolysis occur? Cytoplasm. Where does pyruvate oxidation? On its way into the mitochondria. So basically this is in the mitochondria. Okay. And then electron transport here, we were talking about that earlier. We went back to that. If we go back to this diagram here, the electron transport is on that inner mitochondrial membrane. These are your electron transport carriers here. Okay. And so where does that electron transport occur? That occurs on along, I would write a little note here, along the inner membrane. And not, it includes the carriers within the membrane, but it also includes the matrix space and the inner membrane space on either side, right? So this involves 2 processes. One is called electron transport system or chain, where the electrons are delivered there, and then a process called chemiosmosis. So the rest of the chapter is going to be going through these processes, and then we're going to consider a bit more special topics regarding these processes. So in this section, we're going to look at glycolysis, and recall that the word lysis means to split, and glyco means is reference to sugar. So literally translated, we're going to take a glucose molecule and split it into 2. And so you want to be able to describe the basic process of glycolysis. We're going to avoid details of knowing the chemical structures and so on, but you should be able to interpret glycolysis. What's going on? What are the main things going on within glycolysis? You should be able to understand what the yields are for glycolysis in terms of number of ATPs or electron carriers that are going to go carry high energy electrons to electron transport to make even more ATP later on in the process. And then distinguish between aerobic respiration, which is the use of oxygen, and fermentation. And so what does glycolysis do? Basically, it's going to convert one glucose molecule which is 6 carbons, to 2 pyruvates. So pyruvate is a name you need to know. If we're going to split glucose 6 carbons in half, then you're going to have 2 molecules that are made of 3 carbons. It is a metabolic pathway. This process occurs within the cytoplasm, and it is described as 10 steps. And going back to our picture, here's the mitochondria right here with the other process we talked about in the prior section, and there's glycolysis happening now inside here. And we can see that we're going to actually generate some ATP out of that and some NADH. Okay? So again, it occurs in the cytoplasm. We're going to have a net production of 2 ATPs. Okay, so that's something to remember. How many ATPs? You're going to get 2 pyruvates, 2 ATPs, and we're also going to get 2 reduced form of NADH. So remember, this is going to be carrying those high energy electrons that are taken from the organic molecules, the glucose in this case. And basically, we're producing it from reducing 2 NAD pluses. Okay, so the overall process here can be described as 3 main parts here. And one of them is the cleavage, where we split the sugar from each sphere right there represents a carbon. And we're going to split into 2 3 carbons, and then we're going to oxidize and make some ATP along the way. The first part here, there's going to be a cost of ATP. So this is the priming. So you're going to take the glucose, and whenever you take phosphates and you start attaching them, you're kind of like priming and preparing a molecule to go through further change, just like you're charging it up. And so this is going to cost 2 ATPs. Now you can see over here that ultimately each half of the glucose molecule, on our way to making pyruvates here at the end, each half is going to be producing 2 ATPs. And so if it costs us 2 and we produce 4, then there's going to be a net gain of the 2 ATPs that we see here. So those are things you don't want to look at. So here we're going to have that priming where we have a cost of ATPs. And you can see the phosphates are stuck on either side of that sugar molecule. Then we cleave it, which means to split it into 2 3 carbon sugars. And then you're going to have oxidations. And oxidations, remember, we're going to take off hydrogen. So we're going to have some dehydrogenations. Which are going to be with the help of enzymes called dehydrogenases. And then we have substrate level substrate level phosphorylation for the ATP. There's that diagram again. Here we're looking at this at a little bit more detailed diagram of the process, just to show how it is complex, and there's an enzyme required at every step of the way. We start up here with glucose. Here we have our cleavage occurring right here. Okay. And then we have our oxidation and phosphorylation occurring along either side here, ultimately giving us 2 3 carbon pyruvates. So that's something to remember. You're going to get 2 pyruvates out of this. So we're going to look at this in a little more close detail, just because you should be able to interpret what's going on. Not memorize, but understand where does oxidations occur, where we have phosphorylations happening, and even recognize the name of some of the enzymes based on what we've learned in our prior chapters. Okay, so I'm going to zoom in to the top part there, and we can see right here we have glucose, and right here what we're going to do is we're going to take an ATP and we're going to phosphorylate the glucose. So here's your glucose here, your first molecule here, and we're going to phosphorylate it, and we're going to attach the phosphate here, and we're going to attach it to this carbon here, and that carbon is going to be the number 6 carbon. So that's why it says now glucose 6 for the number 6 carbon phosphate. Okay, and it's done with an enzyme called a kinase. Kinases, they're called kinases in general. Anytime you see the enzyme kinase, it's going to phosphorylate. So they're enzymes that are involved in transferring phosphates from one group to another. So it's a hexokinase because we have a hexose sugar. It's a 6 carbon sugar. And then here we're going to go to another enzyme called an isomerase. Remember, isomers are different. They're going to have the same molecular formula but different structure, so an isomerase. And so what this is going to do is going to take the glucose, so it's a phosphoglucose isomerase, and it's going to make fructose. It's just going to rearrange the atoms. It's still a 6 carbon sugar with that phosphate, and we make fructose 6 phosphate. So here you can see that the 6-carbon sugar looks different structurally. So we go from glucose to a fructose 6-phosphate here. And then we're going to have another kinase. This one's phosphofructokinase, and we're going to add a phosphate to the other carbon, the number 1 carbon. So here we have a number 1 carbon and a number 6 carbon. Remember, there's a way of numbering the carbons: 1, 2, 3, 4, 5, 6 carbon. So now we have a fructose 1,6, and the bis stands for 2 phosphates. So 2 phosphates there. So overall, what are we doing? We are we're phosphorylating, and this is basically the cost part of it. We've used 2 ATP, so we're spending energy doing this. We're priming the sugar to go through change. Okay? And so this is the priming, all of this right here. Okay? So 1, 2, 3 steps, and now we go to the cleavage right here. And so when we cleave, we're going to get 2. If we're going to cut a 6-carbon molecule in half, we're going to get 2 3-carbon ones. And they're not exactly the same. They do have 3 carbons. You can check that 1, 2, 3, and then we have 1, 2, 3, but they're actually different compounds. And so what's going to happen here is we're going to have some isomerase that's going to switch to the same type of molecule. So we're going to go from this molecule here, we're going to form another one of these. So this is called glyceraldehyde 3-phosphate. So we're going to make 2 glycerol 3-phosphates, and then from there, both glyceraldehyde 3-phosphates are going to go through oxidation, and we're going to do some phosphorylation as well. Now remember, each one of these has a phosphate on it right now. Okay? So moving out to the bottom part here, you can see right here, we're going to take an enzyme called glyceraldehyde 3-phosphate dehydrogenase. That's an oxidation. You're going to take off a hydrogen from your organic molecules. Now remember, we're going to have 2 of these. We're going to end up with 2 of those. We have a substrate level phosphorylation, so we add a phosphate to the other side. So now this molecule becomes, we're going to get 2 of these. This one becomes one of those. It goes through isomerization. And you end up with 2 glycerol 3-phosphates, and then we add a phosphate to the end of each one. So each of these is going to have 2 phosphates, okay? And it's these phosphates that are going to be used to phosphorylate ATP. So from this point to this point, you see there's the loss of the phosphate, okay? And where did the phosphate go? It went on to an ATP. It went from ADP to make ATP. So here we've made 2 ATPs. Keep in mind we burned 2 earlier in the priming stage, just a while ago, okay? So we got 2 back. We're at net of 0. We haven't really gained any ATP from that, okay? What else happened? Well, we had a dehydrogenase. That means we need to have lost hydrogen. So if we come back over here and we look, okay, and we see what's going on here, and you count the number of hydrogens, 1, 2, 3, and then this right here, there's 2. So it's 1, 2, 3, 4, 5. If we've lost a hydrogen, then we should see 1 less. So here we go, 1, 2, 3, 4. So there's only 4. There was 5 up here and there's 4 over here. What happened? We oxidized that molecule by taking off a hydrogen with its electron. Where did it go? It went to reducing the NADs. So the NADs, 2 of them, each one gets reduced. The hydrogen that we're adding on there comes from this organic molecule, in this case, glyceraldehyde 3-phosphate. Okay, so we're at this point here. We've taken off a phosphate to make our ATP substrate level. What we're left with now is 2 molecules each, because we're working with the same molecule, 3 phosphoglycerates. Okay, and then we're going to have another enzyme change here, and another change here, and then here we have that phosphoenol pyruvate, which I mentioned earlier as an example here. We still have a phosphate at this point. Okay. That phosphate is going to be taken off of this molecule, that phosphoenol pyruvate, and it's going to be used to phosphorylate ATP. So we're going to have substrate level phosphorylation again, taking ADP and making ATP, and the phosphate came from this molecule here. Okay, and so what's responsible for doing that? Another kinase. There's that word there. It's called pyruvate kinase. And once we lose the phosphate, we're now ending up with 2 pyruvate molecules, which are right here. And each pyruvate is going to have 1, 2, 3 carbons. And this molecule here has lost some hydrogens further back up here because we oxidized. We were taking off the hydrogens with their electrons to reduce the NAD pluses. So Overall, this is that entire process. This is glycolysis. And all you really need to do is be able to look at it and say, okay, I know I'm spending ATP here. That cost me. And I needed to phosphorylate the sugar to prime it. Then we're going to cleave here. And then in this part right here is where some of the important stuff is going on. Here's where we get oxidation. You get NADHs, and you're going to get 2. There's 1 and 2 right there. Okay. Then you're going to get substrate level phosphorylations. There and there. So we end up producing 4 ATPs, but it cost us 2 up here. So your net gain is 2 ATPs here. Okay. Now at this point right here, here's that phosphoenolpyruvate, and you might recognize this diagram right here. This is one that we showed earlier showing that substrate level phosphorylation, and that PEP that's right there is that phosphoenolpyruvate, and it's showing that example of taking that phosphate off of there and attaching it to an ADP here at the substrate level with the help of an enzyme. Okay. What we end up producing is an ATP here, and there's your pyruvate, one of 2 of the ones produced in this process that's going to go through further change here in a bit. Okay, so keeping track now, I suggested that you sketch this picture so that you can remember the structure of the mitochondria, outer membrane, inner membrane, the matrix here in the middle. And we want to remember with our kinesthetic drawing that glycolysis happens out here. We just finished covering glycolysis, where you take a sugar molecule and you end up splitting it over 10 steps to make 2 pyruvates. So you would put a 2 there. How many NADHs did we get? We got 2. And while we made 4 ATPs, we had to use 2, so really we only have a net gain of 2 ATPs from this process. Those ATPs can go anywhere to power anything. Now where are we headed? The next stage, we're going to oxidize the pyruvate oxidation, to make acetyl-CoA enzyme A. Now if we have 2 pyruvates, that's going to basically give us 2 of these, and we're going to get to that in the next section here. But first, we got to look at what happens after glycolysis. If we have oxygen present, okay, this would be aerobic respiration. If you have oxygen present, that oxygen is going to be that final electron acceptor. And where's it accepting it from? It's accepting it from the NADH. Remember, the NADH is carrying this hydrogen with the electron that was taken off of the food molecule, right? That electron has a lot of energy in it. We got to go take that into the mitochondria, to the inner membrane for electron transport. When we do that, that's going to take off that proton and that electron and deliver it there. And what that does is regenerates NAD+, the oxidized form, which can go back and be used for more glycolysis. Glycolysis requires the use of NAD+. Remember, we're going to need NAD+ to make NADH. Okay, this process of taking NADH into the mitochondrion is going to produce lots and lots of ATP, because there's still a lot of energy in that electron that's going to be used later. But what happens though if you don't have oxygen? Oxygen is needed for that final electron acceptor here in the mitochondrion. If we don't have that oxygen, then what's going to happen is we're going to start building up a lot of NADH. And what's going to happen, so NADH is going to build up within the cell. And what's going to happen is we're going to deplete the oxidized form. Okay? Because this process of glycolysis, which requires no oxygen, is going to be using the NAD+ and then producing the NADH, right? Okay. For every sugar molecule that we split into, you're going to end up using up 2 NAD pluses to reduce them to NADH. Over time, you're going to end up not having any of the oxidized form. And once you run out of NAD+, you can no longer do glycolysis, and everything shuts down. You can't produce any more ATP at all. Nothing happens. Okay, so we need a way to get around that. And one way to do that is with a process called fermentation. Now, it's very limited as far as certain organisms being able to do this. Like animals, there's limitations on how much they can do this. We still do this kind of stuff, for example, in our muscle cells, but you can't do this and produce enough ATP with the power of the brain, the nervous system, for example. Other organisms can do just fine for quite a while using fermentation. Fermentation is anaerobic when you don't have any oxygen, when oxygen is not available. Instead of having oxygen as that final acceptor here in the electron transport chain, you're going to have an organic molecule pick up that electron. So the organic molecule is going to take the hydrogen and the electron away to give us back NADH. So at least we can keep running glycolysis out in the cytoplasm and produce our limited ATP. Okay, so that's modeled here in sort of a dichotomy here. There's going along. Here's your pyruvate that we produce at the end of the glycolysis. Remember, we produced 2 of them, right? So just following one of them. If we have oxygen, which is going the downward arrow, okay, those NADHs are going to go and deliver to the electron transport within the mitochondria, freeing up more NAD pluses, and we can continue to run the process. Okay, and if we don't, then we go along this path here, and there's 2 kinds of. So this is the path for fermentation. And there's going to be 2 kinds. There's going to be lactic acid fermentation, which animals do in their muscles. We do this as well. And then microbes, like yeasts, can do alcohol fermentation. And they're producing an alcohol called ethanol. Okay, they're slightly different in process, but the key here is that we need to take that NADH and deliver the electron somewhere to free up and give us more oxidized form, which is NAD+, so we can continue at least to run glycolysis. So what's going to happen here is in lactic acid fermentation, which is one form, okay, the NADH is going to place the hydrogen with the electron onto the pyruvate molecule itself. So the pyruvate molecule is going to be that electron acceptor. So we don't have any oxygens. Where we're going to put the electron, we're going to put it on the pyruvate. So we're going to transfer the hydrogen with its electron onto the pyruvate, and that gives us lactate. Lactate is basically lactic acid, okay? And again, this bacteria can do this, and other microbes, and animals can do this in their muscles. Like we do this in our muscles. If we are overusing our muscles and we don't get enough oxygen in the muscles, we start going through lactic acid fermentation. In microbes, they go through alcohol fermentation over here, and that's going to involve decarboxylation. We're going to lose a carbon from there. So remember, pyruvate has 3 carbons. We produce acetaldehyde, which is only a 2-carbon molecule. It's got hydrogens on there and so on, right? And then what do? We're going to take that NADH and we're going to pass the electron from NADH onto the acetaldehyde, and that produces a 2-carbon alcohol called ethanol, and that's freed up our NAD+. Okay? So these are 2 kinds of fermentation: lactic acid fermentation, and this one is alcohol fermentation. These are ways that basically are a way to regenerate NAD pluses that are necessary to continue to run glycolysis when you have oxygen absent.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
So in this section, we're going to move to the oxidation of pyruvate to produce acetyl-coenzyme A, or CoA for short. And so your learning outcome here is to diagram how the oxidation of pyruvate is going to link glycolysis to the citric acid cycle, which is also called the Krebs cycle that you see there in the diagram. Okay, so we went through glycolysis right now, and now we're going to be focusing on pyruvate oxidation. Now remember that we have 2 pyruvates going through this. Okay. Now if you have oxygen in then the pyruvate will go ahead and become oxidized. Now, The oxygen itself is not directly involved here. Oxygen is further down the events that occur here, but oxygen is required to drive that whole process coming from behind as we move forward. So oxygen is required, and oxidation just means that we are breaking down these organic molecules by removing hydrogens and carbons and so on from there, and removing electrons in the process. So in eukaryotes, which do have a mitochondrion, there is a multi-enzyme complex, which was brought up actually in the last chapter. It was a multi-enzyme complex, which was many enzymes together that will take the substrate and cause it to change, producing an intermediate that gets picked up by the next enzyme within that complex. And this is to make sure that you have control of your metabolic intermediates so that they don't go off and go and do side reactions there. So this one is called pyruvate dehydrogenase, which catalyzes the reaction. In prokaryotes, this process is going to occur at the plasma membrane. In eukaryotes, this occurs as the pyruvate is entering to get into the mitochondrion. We're going to go through this pyruvate oxidation. So keep in mind that we have two pyruvates going through this, right? So each pyruvate is a 3-carbon pyruvate. And what's going to happen is we're going to have a decarboxylation, okay? And that pyruvate dehydrogenase is a multi-enzyme process. So one of the things it's going to do is going to remove one of the carbons. So remember, you had 3 carbons. So now we're going to move to a 2-carbon compound called acetaldehyde, and then we're going to attach another molecule to it called coenzyme A. So then we get that acetyl coenzyme A. And we're going to get 2 of them because each pyruvate is going to give us 1 of those. Okay? Each pyruvate is also going to end up giving us 1 NADH, so we're going to be removing a hydrogen off of the pyruvate during this process. We're going to dehydrogenate, which means we're oxidizing the pyruvate. Remember, when you take the hydrogen off, you're removing an electron, right? So what you end up with is, again, that 2-carbon compound called acetaldehyde, and then it binds to the coenzyme A. So this so the connection here is that pyruvate then becomes the 2-carbon acetyl coenzyme A, which then goes into the citric acid cycle. So the citric acid cycle is another name for the Krebs cycle, and we're going to break down the acetaldehyde further. Okay, so it's also called the Krebs cycle, and it's also called the tricarboxylic acid cycle, because there's 3 main organic acids that are produced, including citric acid. So let's take a look at the diagram here. This is what pyruvate looks like. We saw this earlier. And the pyruvate's going to ultimately form the 2-carbon acetaldehyde. So we see that 1, 2, 3 carbons, you end up with only 2 carbons here because one of the carbons gets taken off with the help of that enzyme pyruvate dehydrogenase. So we lose a carbon in the form, and this is where our first carbon dioxide is produced. Now remember though, it's not 1 pyruvate, it's 2 for every glucose molecule, so that means 2 carbon dioxides, and we're going to end up getting 2 NADHs. So 2 NAD pluses are going to assist in helping to remove hydrogen off of the organic compound. And again, we get the reduced form of NADH. These are electron carriers that are going to go off to the electron transport system there. Okay, so what do we get off of here? 2 NADHs off of the pyruvate oxidation. So in review again, we went through glycolysis in the cytoplasm. Your glucose molecule was split over 10 steps to make 2 pyruvates. We got 2 net ATPs out of there and 2 NADHs. Now, in this section, we covered the oxidation of pyruvate. Now remember, it's 2 pyruvates, so this is going to give us 2 acetyl coenzyme A's. We saw that each one of those acetyl coenzyme A's produces an NADH, so that makes 2 NADHs coming off of there. And we have each one loses a carbon in the form of carbon dioxide, so we have 2 carbon dioxides right there. So in this section, we're going to cover the citric acid cycle, which is also referred to as the Krebs cycle and the tricarboxylic acid cycle. Because there's 3 major carboxylic acid, citric acid, and oxalic acid. Learning objectives here are to relate the nine reactions of the citric acid cycle to the flow of carbon and electrons in the cycle. So we're talking about removing these high energy electrons to use them to help generate ATP in the next part of the process. And then diagram the oxidation reactions in the citric acid cycle. So for this particular objective, I would expect you to just interpret the cycle overall and not necessarily memorize every little detail on there. So what's going to happen here? Well, we had generated the acetyl coenzyme A. That's going to enter, and we're going to have that acetyl group, and it was produced from the pyruvate. It's going to enter the mitochondria into the matrix where we're going to go through that Krebs cycle. This metabolic pathway or biochemical pathway is 9 steps, and there's 3 main parts to those. The first one is the combining of the acetyl coenzyme A and the oxaloacetate or oxaloacetic acid, which when the two combined, they're going to form citric acid. So that's the first step here. So that's going to occur right here. Here you have your acetyl coenzyme A. You lose the coenzyme, and this 2-carbon acetyl group is going to combine with the oxaloacetate, which is 4 carbons. So 2 carbons, 4 carbons makes your 6-carbon citrate, which is citric acid. Okay? So that's the first part. So this is described by the first one. The second one is the citrate rearrangement and our decarboxylation. Decarboxylation means you're going to remove the carbons. They're going to come off as a form of CO2. So that's going to occur from right here. Here's your first carbon dioxide being lost. Here's your second one. And then we're going to come to about this point right here. So this is part 2. And then the last part is the regeneration of the oxaloacetate, which is why it's a cycle. So we're going to go through further changes with the remaining 4-carbon compound. And then ultimately we're going to get back to our oxaloacetate. So along the way we're going to continue to oxidize, and we can oxidize by removing hydrogens and electrons from this organic molecule. Okay. You can account for all of that by looking at the actual structural formulas. There's going to be a picture of the cycle that shows the actual structures of these compounds along the way and their enzymes and so on. Don't memorize the structures though, but just be able to interpret what's going on there. So overall, the description is right here. I encourage you to read through this, but I'm just going to go ahead and describe it. So we've already kind of looked at it overall. Remember, we've done glycolysis already. We did the oxidation of pyruvate. 2 pyruvates were generated from splitting the sugar. Those pyruvates were oxidized to produce acetyl coenzyme A, so each one enters the Krebs cycle, or the citric acid cycle. And this cycle is going to produce more ATP. We're going to find out how many. And it's going to produce more NADHs, or electron carriers, and a new electron carrier called FADH2. And those electron carriers are going to go with oxygen, presence of oxygen, to electron transport chain along that inner membrane. So this step right here is coming up next after this section here. So remember that you're actually going to have what you see in the cycle is going to be times 2, because we had 2 pyruvates oxidized to form acetyl coenzyme A. So acetyl coenzyme A is going to come in here, and it's going to combine with our oxaloacetate. You're going to remove the coenzyme, and you produce your 6-carbon compound, citric acid. That citric acid is going to go through some changes, but you can see here one of the things that's going to happen is you're going to have an oxidation right there and a decarboxylation. The oxidation here is the NAD plus is going to basically carry away a hydrogen and electron from this organic compound here. So here's your reduced form NADH. That's going to be useful. It's going to carry that high energy electron. There goes one of your carbon dioxides. We started with 6, and we now have a 5-carbon compound. We're going to have another decarboxylation, so we're going to lose another carbon. We're also going to remove another hydrogen and its electron to reduce NAD to NADH. And this gives us now a 4-carbon molecule, and it's got a name. And then here we're going to have substrate level phosphorylation. Okay, and so you're going to generate an ATP. Okay, remember this is times 2, so really You're going to go 2 cycles around, so that means really 2 ATPs at this point. Now over here we're going to go through the regeneration, and this 4-carbon compound is not oxaloacetate yet. It's going to go through some changes that include more oxidation by dehydrogenation. So we're going to turn an electron carrier called FAD. It's another dinucleotide, just like NAD was. In this case, it forms an NADH2. And then we're going to oxidize again, a second one. And that's going to give us another NADH here. And ultimately, we get back our oxaloacetate, which completes the cycle there. Now, remember, we're going to go twice around here, so everything is times 2. So this would be 2 NADHs here, 2 carbon dioxides, 2 NADHs, 2 carbon dioxides, 2 ATPs, 2 FADH2s, and 2 NADHs here. So we want to account for all of those ultimately there at the end. So if we look, one complete 2 turns for one glucose molecule overall is going to give us, there's your carbon dioxide, it's going to be 4 CO2s for that one glucose molecule. Remember, we lost the other 2 in the oxidation of pyruvate. For the NADH, that's 2, 4, 6. So 6 NADHs here. And we're going to have 2 ATPs. And we already did that NADH, but we're going to have 2 FADH2s. These 2 are your electron carriers here, so 6 and then 2 there. Okay, so taking a look here, that's what we see here. So this is each turn of acetyl coenzyme A. We already said this is the same diagram here. You're going to release 2 molecules, but we're going to times 2 that, so really this is going to be 4. 3 NADHs, but we came up with 6 because we go twice around for each pyruvate. It says 1 NFADH, but that's going to be 2 for one glucose molecule. This is 1 ATP, but that's going to be 2 for that one glucose molecule, and we regenerate oxaloacetate. This is the actual cycle with the actual formula, so you can follow the losses of hydrogen and so on. So here is your acetyl coenzyme A. It's got 2 carbons here, and you're going to lose the coenzyme. And it's going to combine with oxaloacetate to form your citrate, which should have 6 carbons. 1, 2, 3, 4, 5, and then 6 carbons there. It's going to go through a change called isocitrate. It's still 6 carbons, and then we have our decarboxylation here and an oxidation here where we remove a hydrogen. So if we were to go and count all the hydrogens here on isocitrate, we should have 1 less hydrogen here in alpha-ketoglutarate. Alpha-ketoglutarate, remember that because we're going to see that again in a little bit. But that's one of the intermediates there. And if we went and we calculated the hydrogens, you're going to see 1 less at this point here. Why? Because we took it off of the citrate to reduce NAD, to make NADH. So here's an oxidation right here, right? What are we oxidizing? We're oxidizing the organic compound in order to reduce the NAD, and we reduce it by making NADH, okay? And then we have our other decarboxylation here and dehydrogenation / oxidation. So if we go from alpha-ketoglutarate to succinyl coenzyme A or succinate here, you're gonna have 1 less hydrogen overall. Then we have substrate level phosphorylation where we generate one of the ATPs. Remember, this is just 1 turn. We got to go twice, right? Then we have our succinate. That's going to be going through an oxidation, so we lose another hydrogen from here, and that gives us fumarate. Here's where we generate our FADH2. Water comes in here. When that water comes in, we're basically going to add some hydrogens here, so there's a sort of a reduction of fumarate to malate, and then immediately we go through another oxidation. So we lose another hydrogen to produce NADH, and then ultimately that gives us oxaloacetate. So every time you see the production of NADH or FADH, you should see less hydrogens on there because you're oxidizing them. You're removing the electron with the hydrogens. Remember, that's always the case, right? When it comes to this organic compounds being oxidized, we're going to remove the hydrogen and we're going to add it to some other compound. In this case, we're going to add it to our NAD+. And so that gives us NADH. The electrons come with it, and you're going to have your oxidized R without the H. Okay? And so you should see we've lost that. There's other hydrogens on the R. It's a bigger it's a compound with other atoms in there, but it's lost a hydrogen during that oxidation. So let's go back to our picture again and let's review. Remember, for glycolysis, we start off with 1,6-carbon glucose molecule. At the end of the 10 steps, you get 2 pyruvates. We had 2 NADHs produced there. We had 2 ATPs produced. The 2 pyruvates enter. Both of them are going to be oxidized. Pyruvate oxidation, that's going to give us 2 acetyl coenzymases. That process produced 1 carbon dioxide for each pyruvate, so that's 2 carbon dioxides there. And each one of those produced 2 NADHs, so we have 2 NADHs right there. Okay. Now then, we went into Krebs cycle, the citric acid cycle, right? In the citric acid cycle, for each 2 carbon acetyl coenzyme A, we lost 2 carbon dioxides. They got removed, decarboxylation. But that's 2 for each one of these, so it's 2 times 2, so that gives us 4 carbon dioxide. So go back to that NADH2. How many did we produce? There was, from memory, there was 1, 2, 3 going around that circle times 2. That's a total of 6 NADHs there. There was 1 FADH2 for 1 turn, so 2 turns is 2 of those there. And for the ATP, during 1 complete turn, you produced 1, but we go twice for each of the pyruvates that came in. So that's 2 ATPs. Okay. So let's add them up. How many carbon dioxides do we have altogether? The oxidation of the pyruvate to begin with, and then the citric acid cycle, that's 4. That's 6 carbon dioxides. Okay. That matches the carbons came from that glucose. There was 6 carbons there. Okay. Total ATPs to this point. We had 2 here, net of 2. 2 here. So that's a total of 4 ATPs only so far. There's a lot more ATP we got to make. Okay. And then how many NADHs altogether? 2, 4, and 6 more. That's 10 NADHs. And 2 FADH2s. There's still a lot of energy here in the electrons that these guys are carrying. And that energy, we can see these arrows. These NADHs and FADH2 are going to carry it to the electron transport chain, where we're going to generate most of the ATP that's still available to be made, or that we can still make. Okay, so let's check. We came up with 6 carbon dioxides on that list. We had 4 ATPs. We had 10 NADHs and 2 FADH2s. Check, 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 check. We got it. Okay, so the idea here is at a most fundamental level, guys, for this course, you need to tell me where glycolysis happens, what are the main products from there. And if I give you some information, you'll be able to tell me if an oxidation is occurring and how that occurs. Remember that anytime an NAD or an FAD, we add hydrogens to them. You're also adding electrons. So what we're doing is we would be reducing these, and what are we oxidizing? We're oxidizing the organic compound, the food molecule, okay, as we break it down. At this point here, we've removed electrons from the food completely, and they're being carried by these guys right here, okay. We've released about 53 kilocalories per mole of sugar that we've broken down, or glucose, and it's been done in steps. We didn't do it all at once, or we're going to just blow up the cell. Okay. And so what are we going to use that energy from that's still tied up here? Well, we made some ATPs, but that's nothing. We got a lot more to make, and that's going to be an electron transport.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
So this section covers the electron transport chain and chemiosmosis. Your learning outcomes here are to describe the structure and the function of the electron transport chain, and then diagram how the proton gradient connects the electron transport to the synthesis of ATP. So electron transport is going to be a series of electron carriers that are going to be found in the inner membrane of the mitochondrion. I showed you this earlier during, I think, the first section of this chapter here. You have your outer membrane, and then along this inner membrane, all along it, you're going to have these carriers. In the image I showed you, was we had that inner membrane here, and we were modeling it from the top part in the picture there. In this case, they're looking at the bottom part of that inner membrane down here, and that's what you see right here. So if that's the case, then the inner membrane space is right here on this side, and the matrix of the mitochondria is on the top side. This is opposite of the picture that I showed you earlier. So you can see these complexes here built into the bilayer, phospholipid bilayer of the inner membrane of the mitochondrion. And there's several of them there. And the basic idea here is that those electron carriers that we saw that were made during glycolysis, during the oxidation of pyruvate, and during Krebs, we had NADH produced, and then Krebs had, unique to it, had FADH2 produced. Those hydrogens on there have associated with them those electrons that were taken from the organic food molecules. And they still have a lot of energy in them, so we're going to come and deliver them. The NADH, if we were to just say, simplify this and just say one series of these carriers, and there's many of these combinations throughout the entire inner membrane, and it's folded to provide a lot of space for this to be happening on a high level, as many of them are working all at the same time. These chains, let's say we're moving along in this one direction, the NADH is going to deliver very early in the chain. And so those electrons basically it's like delivering them higher up on that staircase. So the electrons associated with NADH are basically going to have higher energy because they're higher up on the staircase. The FADH is not going to deliver until we get past one of the first major carriers. So here's where the FADH is and delivers there. So the FADH is delivering a little bit lower on the staircase and has less energy associated with it. But essentially, as these electrons are being delivered, we're basically reducing the carrier, and then the carrier transfers those electrons to the next carrier, and the carrier becomes oxidized while reducing the next one, and so on. At the very end, we have oxygen waiting down here to receive those electrons. So as we're doing this, these carriers are sometimes carrying protons, and what they're doing is they're carrying them against their concentration gradient, and that means that requires energy. So what we're doing is basically taking protons from the matrix and concentrating them at a high level in the inner membrane space, that space that's right here between the two membranes. And that's going to create this great deal of potential energy that we can use to make ATP. And so what we're going to, and we could refer to those proteins or those complexes that are within the membrane, those carriers, we can refer to them, the ones that are going to move the protons, as proton pumps. Where are they getting the energy from those high energy electrons from the carriers NADH and FADH2. So just as a comparison, This is the image that I showed early on during the first section of this chapter here. And the intermembrane space was on top and the matrix was on the bottom. And those were your electron carriers through this chain. And they were pumping the protons up on this picture here. The picture from your book has this flipped upside down because instead of being on the upper part of the membrane right here, we're on the lower part here. So the intermembrane space is below over here. So going back to that image there on the top right, it would be like if we flipped it upside down. These are your carriers here. These are our carriers here. And then we have our ATP synthase here. This is our ATP synthase right there. Okay, so we're going to focus basically on the one that's produced from your textbook here. And so your electron transport chain is on that bottom bilayer here. Here's your NADH. Is bringing that electron that it picked up either in the Krebs cycle or glycolysis or oxidation of pyruvate. Remember, NADH was produced in these parts here. And it's delivering the electrons here and releasing those protons. So the protons are freed, they're in the matrix. And as the electrons are delivered here to this one, we are basically reducing the first carrier. There goes your NAD plus, and it can go back to glycolysis or Krebs to go and pick up more electrons there. But as the electron is moving through this carrier and then gets passed on to the next little carrier there within the membrane, we're basically pumping a proton across from the matrix over here, and we're building up protons on this side here. And this is the intermembrane space. As we move here, FADH2 is delivering its electron there a little bit later, so a little bit lower energy. And those electrons, doesn't matter where they came from, are going to move to the next complex here. They do have some names to them, like B1 complex and cytochrome oxidase complex. Don't have to worry about learning the names of these complexes. But again, we reduce this carrier, and then it becomes oxidized as we transfer the electron this way, pumping more protons, and then we pump more protons here. At the very end over here, we have oxygen waiting to pick up those electrons, along with 2 protons from the matrix there. 2 protons are basically the nucleus of hydrogen atoms with the 2 electrons. When we add them, we get water, which was mentioned earlier. So this is your metabolic water produced by the metabolic actions that have been going on. And so water is produced here. So your final electron acceptor was that oxygen, and technically it's half an oxygen for 2 electrons coming in to make 1 water molecule. Now all of these protons that have been built up here are going to build up this concentration gradient, and ultimately they're going to have a hard time getting through the membrane because the inside of that membrane is hydrophobic. So basically they have a better route. So they can't get through there, but what they can do is they can get through this this protein complex that's partially embedded in the membrane, and it's called ATP synthase. And so the energy of those protons, the kinetic energy of those protons moving through there, is going to provide the energy necessary for taking ADP and inorganic phosphate and phosphorylating to give us ATPs here. And a lot of ATP is generated by this. So this is called oxidative phosphorylation, associated with that oxygen acceptor there at the very end. So when that buildup of protons, that concentration gradient that's been produced of these protons in the intermembrane space are allowed to move through the ATP synthase, that process is called chemiosmosis. Kind of sounds like osmosis, but it's chemiosmosis. And here, this energy, this potential energy that is now being put into motion to go through that ATP synthase, again, is what's going to be used to generate ATP. And we can see here, they provide a number there. So up to about 28 ATPs are produced by those carriers through this process of oxidative phosphorylation. Just before we got here, we only had 4 ATPs produced during the whole time, right? There was 2 produced here and 2 produced here for each glucose molecule. 28 more were able to be produced by the energy that was still carried by those carriers that brought to the electron chain. And that energy was indirect through the process of chemiosmosis when we picked up and increased that concentration gradient, pumping lots of protons in here. When they're allowed to go back through the ATP synthase, that's where we generate the ATP. So know that term, chemiosmosis. It's connected to the electron transport. So now we've come up now total 28 plus 2, 30, and 32 ATPs for one glucose molecule. So looking at the ATP synthase itself, that enzyme there, it's actually, again, that proton gradient. We have that buildup in that intermembrane space, and then they flow back through the enzyme into the mitochondrial matrix. There's two parts to it. There's the part that's embedded within the membrane. It's called the F0 complex. And it actually works like a little motor, and it rotates. And as it rotates, it's rotating because of the energy of those moving protons going through there. As it rotates, it changes the shape of the F1 complex, and it changes it into a conformation that activates it. So it's in the inactive form, and then as the protons are moving through there, its conformation changes, the shape changes, and that is going to activate it and allow the ADP and the inorganic phosphate to be combined through the enzyme action of this ATP synthase. And so this phosphorylation, where we're putting that inorganic phosphate onto the ADP to make ATP, this one is called oxidative phosphorylation. It's different than the ATPs that were generated during glycolysis and during Krebs cycle. That was substrate level phosphorylation.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
In this section, we're going to look at how much energy we get from aerobic respiration. So here, we should be able to have some sort of a theoretical understanding of the number of ATP molecules that are produced by aerobic respiration per molecule of glucose. And we could scale that up to per mole if we want. It's just the same numbers, but in moles. So 1 mole of glucose would give you the same number of ATPs, but in moles. So the theoretical yields for ATP from oxidizing one glucose molecule for bacteria would be 32. It is theoretically 2 less for a glucose molecule in eukaryotes. And part of the reason for that is that it costs a couple of more ATPs to get that pyruvate into the mitochondrial membrane. So we start to look here at where the ATPs came from. So remember, ATPs are generated during glycolysis. We get 2 of those. That's there. We also get 2 ATPs per molecule of glucose from the Krebs cycle. The other ATPs are going to be generated by oxidative phosphorylation. And that's due to the electron carriers taking electrons from the oxidation of the organic molecules. We got 2 NADHs coming from glycolysis, 2 from the oxidation of both pyruvates. These are the numbers for 1 full molecule of glucose, right? 6 total NADHs from Krebs cycle and 2 FADH2s. So when we look here at the ATPs produced from chemiosmosis here, these 2 NADHs give us 5 ATPs. 2, we get 5 more. Here we have 6 NADHs, gives us 15. So we start to look at the ratio here. If we do, we get 5 ATPs for every 2 NADHs. That's about 2.5 ATPs produced per NADH. Okay. That ratio is still the same here, 15 for every 6. 15 divided by 6 is going to give you 2.5 as well, so it's the same ratio. So you get about 2 ATPs per NADH. For FADH2, it's less. The ratio is 3/2, which is about 1.5 ATPs per FADH molecule. Either way, these are the absolute numbers all the way through, including the ATPs produced right here by substrate level phosphorylation, and these are the oxidative ones, the chemiosmosis, oxidative phosphorylation, and we get our total of 32, 30 for eukaryotes because it costs overall. So when we start to look at efficiency, they start to look at, you can look at theoretical calculations based on the amount of ATPs produced for the amount of oxygen used. So it's the phosphate to oxygen ratio, right? And so the bigger that ratio is, the more efficient you would be. The lower the ratio, the less efficient it would be. So we can use these ratios and theoretical calculations and then compare them to actual yields in real living situations, real systems, living systems. And so the numbers will vary. So just remember around the low 30s or so. Sometimes you might see a book report 36, 38 ATPs altogether. So remember in the low 30s and you'll be okay. There's a lot of debate on this. It's just because people are getting different actual experimental values, and they don't always match the theoretical yields. So we're going to look now at how aerobic respiration can be regulated. And you want to, for the learning outcomes here, we want to understand the control points for cellular respiration. So this is going to be done by feedback inhibition, something we talked about in the prior chapter. And there's 2 key control points. One control point is in glycolysis, the enzyme phosphofructokinase, which is going to help phosphorylate the fructose phosphate molecule. And it's allosterically inhibited. Remember, allosteric enzymes have 2 conformations, and in this case, when in one conformation the enzyme is active and the other one is not, so it's going to be inhibited. So it's sort of a negative feedback. It'll be inhibited when you have good amounts of ATP. So the ATP itself would work as an allosteric inhibitor, and also the molecule citrate, which we find in the citric acid cycle. So here we have citrate here. And if it builds up, we can see it comes back here in this little vertical line there. It means inhibits. Same thing, if you have enough ATP produced, it'll come back and inhibit this enzyme. And when it's inhibited, it slows down the progression of glycolysis. So you'll have less of the rest of glycolysis happening if you inhibit a key enzyme in that process. The second control point is in the pyruvate oxidation and citric acid cycle. So there was an enzyme, remember, pyruvate, called pyruvate dehydrogenase, and that would be inhibited by high levels of NADH. So if you have high levels of NADH right here, that would go back and inhibit the enzyme that basically oxidizes the pyruvate, which is that first step before going into the Krebs cycle. So we would have less acetyl-coenzyme A, and that would slow down the entire process there. And then you also have the citrate synthase would be inhibited by high levels of ATP as well. So the citrate synthase would be the key enzyme that's right here at this point, where acetyl-coenzyme A or acetyl-CoA is going to come in here and bind with the oxaloacetate, which is in the Krebs cycle, binds with it to make citrate. So if you have good amounts of ATP, that would inhibit that enzyme that's about right there in the process. So that's all negative feedback inhibition, and it's due to the enzymes being inhibited by the accumulation of these products in the process. So here we're going to talk about oxidation without oxygen. So here we're talking about anaerobic as opposed to aerobic. So here we should be able to compare for your learning outcomes anaerobic with aerobic respiration. We just finished covering aerobic respiration and then distinguish between fermentation and aerobic respiration, and both are without oxygen. So we look at number 1, 2, here's anaerobic, and then 2 is fermentation. So for anaerobic, you're going to use inorganic molecules. That's the difference. Fermentations, you use organic molecules as your final electron acceptor. And oxygen is considered inorganic, but we're talking about inorganic other than oxygen. Okay? Many prokaryotes, which are your bacteria, are going to use inorganic compounds like sulfur, made from sulfur nitrates, carbon dioxide, and even inorganic metals can be used as your final electron acceptor. Again, for fermentation, it's going to be organic molecules as the final electron acceptor. We already talked about it earlier. We briefly mentioned there was lactic acid and alcohol fermentation. So looking at anaerobic respiration, we're just going to look at a couple of types here. There's a group called methanogens, which literally means to generate methane. And methane is a molecule that structurally looks like this. It's the simplest organic molecule. It's CH4, and structurally looks like this. Here, so basically for methanogens, a molecule like CO2 inorganic is going to be the final electron acceptor. It'll be reduced as we put hydrogens on there with the electrons to produce the carbon itself to produce CH4. This type of metabolism is found in diverse organisms, but we're talking about microbes. It says including cows, but not the cow itself, the animal tissue, but within the gut of the animal, there's microorganisms in there that can produce methane by anaerobic respiration. Then you have sulfur prokaryotes, and here they're going to use inorganic sulfur, for example, the sulfur oxide here, and then reduce it to form hydrogen sulfide. So here that sulfur is reduced, and it's reduced because we're putting hydrogens on there with the electrons. And these sulfate producers were important in the early stages of evolution of photosynthesis. The early photosynthesizers didn't produce oxygen, but they used compounds like this as sources of electrons. It'll make more sense when we go over photosynthesis later. This is just an example of a place where you might find some bacteria living in extreme environments. In this case, these bacteria that use sulfur as their final electron acceptors. And then we have fermentation here. This is something we already covered. We have two kinds, general kinds. We have alcohol fermentation and lactic acid fermentation. And in both cases, we've got glycolysis occurring here. We're breaking down sugar through the steps of glycolysis we covered earlier in the chapter. Remember that glycolysis produces a net of 2 ATPs, and the end product is 2 pyruvates. Each one of them looks like this molecule right here. And it's the same for both. The top one is alcohol fermentation, the bottom one is lactic acid. When we get to this point, we're at the same place. We have 2 pyruvates. We produce 2 ATPs. This happens in the cytoplasm. And then here's the difference here. For alcohol fermentation, we're going to have a decarboxylation. So we're going to remove one of the carbons. That's 3 carbons. So we remove it. We get a 2-carbon acetaldehyde. There's your 2 carbons there. And then that NADH, because there's no oxygen, it cannot go deliver the electron that it's carrying with that hydrogen. It can't go deliver it at electron transport because there's no oxygen there at the end waiting to pick it up. So that means we're going to have an accumulation of NADH, and we're going to have a reduction of NAD, the oxidized form. So what we want to do is this metabolism allows us to move that electron onto the acetaldehyde, basically reducing this molecule. And when we reduce it, we end up forming ethanol. Okay? So you count up the hydrogen, you see you're going to have more hydrogens on here and your ethanol. But what that's done is it's freed up NADH to allow us to continue to do glycolysis. It's not efficient because remember most of the ATP is made during oxidative phosphorylation. But at least we're making some ATP here, and we're able to continue to make 2 ATPs for every sugar molecule we break down. The thing is, there's still a lot of energy left in that pyruvate. We just can't tap into it. For lactic acid fermentation, so alcohol fermentation can occur in a variety of microorganisms, including yeasts and bacteria, certain bacteria. Lactic acid fermentation, also microorganisms can do it, but we also in the animals and in muscle cells would do lactic acid fermentation. And here, the main difference is we're not going to have that decarboxylation that we had in alcohol fermentation. So we're going to keep the 3 carbons, and what we're going to do is we're going to reduce the pyruvate directly. And when we reduce the pyruvate, we make lactate, which is basically lactic acid, right? So remember, it's going to be 2 pyruvates, 2 NADHs are going to be used to reduce it, and that frees up the NAD to continue to do glycolysis. Again, there's still a lot of energy in that molecule. It could be oxidized a lot further to produce more ATPs if oxygen was present. So these are the 2 examples of fermentation to know. So here in this section, we're going to look at how we might use other molecules other than glucose as a way of generating ATP. So catabolism, remember, is the breakdown. And so when we break down other biomolecules like proteins and fats, we can also generate some ATP. Now, proteins is not the primary way we want to generate energy, and fat is a great way of storing lots of energy for this. So how do we go about accessing these compounds to generate ATP? So what we want to do is, for your learning outcomes, is identify how proteins and fats can enter those metabolic pathways during energy metabolism. So at what point do they enter during glycolysis? Do they enter during Krebs? And then recognize important key intermediates in this sort of metabolism. So here you have an amino acid called glutamate. And amino acids, remember, they have this central carbon, they have the amino group, and they have the carboxyl group, and then they have an R group, side chain. And the key here to entering the metabolic pathways for generating ATP is to remove the amino. It's called deamination. And so when we do that, we remove the amino group and it becomes ammonia, and then it may be converted to urea, like in our system, for example. Urea ends up in our urine, is filtered from our blood, and then we eliminate it with the urine. But glutamate, the deamination of glutamate produces alpha ketoglutarate, which earlier in an earlier section said, remember that compound. Remember that as part of Krebs cycle. You find it in Krebs cycle as we are decarboxylating during that process, the decarboxylation and oxidation. And so this compound can enter Krebs cycle and then go into that a biochemical pathway to help produce more ATPs. So the amino acids, the amino acids, when they do this, and they give this example of glutamate, they have to have their amino group removed. That's called deamination. Okay, and that's where you get your metabolic waste. So when we are metabolizing proteins and breaking them down, that's where the nitrogenous waste can come from. And so the products can enter in various parts of these metabolic pathways for generating energy. Alanine can be converted into pyruvate, for example. Remember, then pyruvate can be oxidized to produce acetyl-CoA enzyme A and enter Krebs cycle that way. Aspartate is another amino acid. That one can be converted to oxaloacetate. That's one of those key parts of the Krebs cycle. This is that one where the acetyl-CoA enzyme A comes in and then combines the acetyl group with the oxaloacetate to make citric acid. And so that's just looking at deamination process. And then we look at fats. So the amino acids can be broken, deaminated, and then enter in various parts. When it comes to catabolism of fat, remember the fatty acids. The fatty acids will vary in length and whether they're saturated or unsaturated. But remember that the fatty acid itself has a carboxyl group right here, and then a chain of hydrocarbon chain, long hydrocarbon chain. And so the fatty acids are going to be converted to acetyl groups by a process called beta oxidation. And this is an oxygen-dependent process, so it's an aerobic process. And we can see here that the process overall is going to every 2 carbons going to allow us to generate an acetyl coenzyme A, which can enter Krebs cycle. So this goes through that process here. We're going to need to use ATP. Coenzyme A will bind right here. And then remember, there's going to be more carbons out here. So there's some variable chain length of hydrocarbons. And we're going to go ahead and go through some of that oxidation here. So we have dehydrogenation. And so FAD is going to be reduced to FADH2, so we're taking hydrogens off of there. We've attached our coenzyme A. We're going to add in a water molecule here and another dehydrogenation. And then ultimately we break off 2 of the carbons here, which is going to give, with the coenzyme A, is going to give us acetyl coenzyme A. And what we have left repeats. So every time we repeat, we're going to be 2 carbons short. So this entire process here is called beta oxidation. We are oxidizing the hydrocarbon. We're removing hydrogens from there. We're adding them to FAD and NAD. And remember, these guys here are going to go off and can go off to electron transport, taking those electrons there. And then the acetyl coenzyme A can go through their Krebs cycle and produce more. Each one can produce 3 NADHs and 1 FADH2 once you're in the Krebs cycle. So this is a repeating process as you go down, and you're removing 2 carbons every repeat of this process. Now, overall fats, fatty acids are going to have a lot more energy in there because they have a lot of carbon-hydrogen bonds in there. And so if we compare this, just 6 carbons on this fatty acid chain to the 6 carbons in glucose, right? And so fatty acid is going to have a lot more carbons on the fatty acid, these chains of hydrocarbons. But if we compare every 6 carbons that we get off here, so basically going through this process 3 times, you're going to get 20% more energy in that molecule than you would from a 6-carbon glucose. So fats are a really great way for long-term storage of energy to produce ATP. So when we look at this overall process, basically you can metabolize any one of these biomolecules in a way to where we can enter some point in the energy metabolism pathways. So we've already looked at sugars, polysaccharides, like glycogen or starch. We can break down to simple sugars, go through glycolysis, turn to pyruvate, acetyl coenzyme A, and then run your citric acid cycle, produce ATPs there. Your product's going to be carbon dioxide. Water is also metabolic water is produced. When it comes to proteins, we can break them down to amino acids, deamination, and then we can enter at various points in these metabolic pathways. Even nucleic acids can be deaminated and the products there can enter some aspect of, say, the citric acid cycle. And every one of these processes is reversible. So you can see the arrows going back the other way over here. And this brings up a point here is that where do you get your, a few points here. Where do you get your nitrogenous waste? It's going to be from the metabolism of proteins, the catabolism of breaking down, and of nucleic acids, which we find in RNAs and DNAs. And over here, if we look at this point here, the sugars here. So the sugars we can reverse back and store glycogen or whatever. And when we have a whole bunch of sugars here, we can enter into the citric acid cycle. And then the fats, remember the fats can be broken down by beta oxidation to produce acetyl coenzyme A. But both the sugars, even proteins, but thinking about sugars and diet and so on, what we eat, what we consume, the sugars can be oxidized down to pyruvate and acetyl coenzyme A. And if we have no need to produce any more ATP, we're not doing any aerobic work, no aerobic exercise, we're going to have a buildup here. And what's going to happen is that acetyl coenzyme A can now be reversed in this direction here to produce fatty acids and then fats. So excess sugar that's not going to be used up, that basically can be taken into long-term storage as fats overall. So that's interesting that every one of these biomolecules can be used to generate energy ATP, but the quick source is going to be sugars and long-term storage with lots of energy that can be used for also generating ATP are these. So these are your two primary energy nutrients, and then we use proteins and nucleic acids as they might be needed. This last topic considers the evolution of metabolism. And so your learning outcome is to describe one possible hypothesis for the evolution of metabolism. So if the idea that life has not always been on this planet, and then you have the beginning of life, then you have to have some sort of metabolisms that evolve as that with that life. Because the process of life are the reactions that occur within these organisms, these cells. So a hypothetical timeline, using a little bit of logic and clues from certain lines of evidence, one possibility is that we had to have some sort of an energy storage molecule. So we use ATP today, might have been ATP then. And then one of the most ancient metabolic pathways is glycolysis. One of the clues for that is, the oldest pathways is that all living organisms are capable of doing glycolysis, from eukaryotes to prokaryotes. So it is a pathway found in all living organisms, and that means it's been conserved this whole time. It's that important. And then we had anaoxygenic photosynthesis. This means without producing oxygen. We know photosynthesis, familiar with plants doing photosynthesis, they produce oxygen. But here, and by the way, oxygen comes from doing the opposite of what we just covered during aerobic respiration. Oxygen is there at the end to capture those electrons, and with some hydrogen to produce water. Photosynthesis does the opposite. It splits water, removing those electrons. And so that's a hard thing to do, but they use energy from the sun. So in the early metabolism, photosynthesis, they weren't splitting water. They were the photosynthetic organisms were splitting H2S instead of H2O. And so what that would have produced instead of oxygen is sulfur. So they were generating sulfur as a product. So the early photosynthesizers were anoxygenic. And then we have the evolution of oxygenic photosynthesis. So here we're not using dihydrogen sulfate. Instead, we're using dihydrogen monoxide, which is water. And oxygen is being produced, and that basically changes the atmosphere of the Earth at that point. So before that, there was no oxygen in the atmosphere. All the oxygen was bound to within compounds, perhaps carbon dioxide, iron oxides like rust. And then the next thing to evolve, we had to have a good way of producing compounds that contain nitrogen that can enter living systems. Nitrogen in the atmosphere is N2, and it's very stable because nitrogen itself has a triple bond there. And so this would be the structural formula for nitrogen. It's pretty hard to break those 3 bonds. They're very strong. But if we could have organisms that take nitrogen from the atmosphere and fix it into compounds that can then be taken up by organisms, because nitrogen is needed to make nucleic acids and to make amino acids that produce proteins. So these are major biomolecules, and we need good source. So we have to have the evolution of that nitrogen fixation. And then probably one of the most recent metabolisms to evolve was aerobic respiration. It couldn't have come first because the rocks that tell the story, these ancient rocks, show that there was no oxygen in the atmosphere. And so you had to have oxygen first, oxygen production first for metabolism that would take advantage of that availability of oxygen as an electron acceptor. So this is a hypothetical, a hypothesized timeline. Metabolisms evolved.